Howdy, my name is Adamari Cerda, and my partner Mario Flores Castro and I will be discussing our ninth experimental lab report on the phage experiment. To begin, bacteriophages are a subset of viruses that infect bacteria. These microorganisms provide insight into host infection mechanisms that enable applicable methods relating to bacterial control and genetics. This investigation focuses on culturing and identifying potential bacteriophages that may be applicable to future use. The materials needed for this investigation include a 10 to the negative 1 dilution of phage, 4 LB plates, 9 microcentrifuge tubes, sterile water, micropipettes and their corresponding tips, 4 large test tubes, Escherichia coli in a broth, and the unknown gram negative 1 sample. For the first part of the investigation, we will focus on creating serial dilutions of phage in sterile water. First, we will obtain the 10 to the negative 1 dilution of phage and label 5 microcentrifuge tubes as follows: 10 to the negative 2, 10 to the negative 3, 10 to the negative 4, 10 to the negative 5, and 10 to the negative 6. Then, we will add 900 microliters of sterile water to each of these new tubes. From the 10 to the negative 1 tube, we will then obtain 100 microliters and add into the 10 to the negative 2 tube. Then we will mix well. Additionally, from the 10 to the negative 2 tube, we will obtain 100 microliters and add into the 10 to the negative 3 tube before mixing well. Then we will repeat until the 10 to the negative 6 tube and ultimately discard the extra 100 microliters. Part 2 of this investigation involves creating a whole plate titer from the serial dilution. First, we will label one LB plate with the type of phage and dilution used. We will then obtain and label 4 microcentrifuge tubes, E3, E4, and E5. From the original dilution tubes prepared in part 1, we will then aliquot 100 microliters into each of the E tubes created. Then we will aliquot 100 microliters of the E. coli stock into the phage containing dilution before vortexing. Then those tubes will be allowed to sit for 10 minutes. Part 3 of this investigation calls for plating the phage dilutions. First, we will obtain 200 microliters of the phage and bacteria mix and add them into a test tube. 5 ml of molten top agar will be added and then immediately poured into the plates. The plates will then be swirled to make sure they are spread out evenly. Then they will be left undisturbed for the lawn to set. Last but not least, these plates will be sealed with parafilm and left to incubate.
And these were the results of our bacterial plates after incubating. As we can see, there is bacterial growth present, and there's the presence of plaques, which indicates that the phages was successfully isolated and grown of the bacterial plates. However, we saw that the dilutions were not enough, as 10 to the negative 3, 10 to the negative 4, and 10 to the negative 5 had too many plaques to count. However, 10 to the negative 6 showed that there was 165 plaque forming units obtained. We used that and then divided it by the dilution factor times total volume to find the plaque forming units per milliliter. We found that that was 8.25 times 10 to the 8 plaque forming units per milliliter. This means that for every milliliter, there's 8.25 times 10 to the 8 phages. In conclusion, the black assay revealed a high concentration of bacteriophages in the sample. Some of the dilutions show plaques to many to count. However, 10 to the negative 6 had 165 plaque forming units, with a concentration of 8.25 times 10 to the 8 plaque forming units per milliliter. These findings offer insight on how plaque assays are a powerful tool when quantifying phages and their density in a sample. Thank you.